Good afternoon and welcome to the Winter Spring EM course here at the Simons Electron Microscopy Center. Our guest lecturer today is Tom Walz from Rockefeller University. He has a long career in electron microscopy. He has done several different modalities of EM from 2D crystallography to single particle, and that he's been working on very challenging systems that include memory proteins and macromolecular molecular complexes and lipid environments. So Tom, welcome, and thank you for joining us today.
My pleasure, I guess. It's a new format for me, so I'm gonna get used to this, I guess.
Okay, I'll put down the title slide so you can get a better look at everyone in the class.
Appreciate it.
So given that you've been in the field of EM for a long time, how did you first get into electron microscopy, and when did that shift to cryo electron microscopy for you?
So basically, I got into EM when I was trained as a student in the Biozentrum in Basel, Switzerland. And the way it works in Switzerland is you have kind of block courses where you for several weeks are focused on particular themes like biochemistry, cell biology. And there was this little block course that was focused on electron microscopy. And the reason for that was that one of the pioneers in electron microscopy was Edward Kellenberger, who was working on virus assembly. And so he basically, the Biozentrum was an early center for electron microscopy. And so in this block course, we basically put a drop of water, which looked like water, onto the grid. And then when we looked at it, it was full of T4 phages. And I was just fascinated how these T4 phages, which look like little moon landing capsules, how they managed to assemble themselves into these very complex and intricate structures. And with this, I was actually hooked. I really wanted to understand self-assembly and use electron microscopy to do that. And so when I was ready to do my diploma thesis, I approached Kellenberg and asked him whether I could join his group. But he was already on his way out. He was close to retirement, and so I wasn't able to do that. And then I tried to join a different group which used electron microscopy to look at integrins. And there were two people, and we flipped the coin. I lost, so I did not work on integrins. And so I approached a third group, which was Andreas Engel, and he used two-dimensional crystals to look at membrane protein structure. And he did accept me, and so that's how I ended up with electron crystallography and looking at membrane protein structures. And in the Biozentrum, we did mostly just negative stain EM. We were not equipped at that time. That's many, many, many years ago by now. And at that time, we were not equipped for Cryo EM. We didn't have instruments that could do Cryo EM. So when my samples, my 2D crystals became good enough, I had to go to different places to do cryo. And so I actually did learn Cryo EM with Wolfgang Baumeister, a guy called Dieter Tübke, in his group. And while during the day that the tomography, Dieter and I were sitting through the nights trying to collect images of our 2D crystals. So, but the problem was that Dieter Tübke was only theoretically working with 2D crystals. He never really did it. He was also an electron tomographer. And so when the crystals really became very good, I ended up going to Japan to work with Yoshinori Fujiyoshi, who developed these top entry helium-cooled microscopes, which at the time were really the best microscopes. And so that's where I really collected images and diffraction patterns of 2D crystals. So that's how I got into Cryo EM.
Do you have a favorite microscope, or what was your first microscope?
My favorite microscope is a CM10. And the reason is that I can still use it. And it's an old, robust instrument that still has all the knobs and whistles, and you can just go and crank some knobs and change the focus. And these days, everything is automatic with computers, and it's much less intuitive for me to work on those instruments. And so CM10 is basically instant gratification. You put your sample on a grid, you stain it, and you put a microscope, and you immediately see what it is. So to me, to this day is just the most interesting part, because it's the first time you actually see your protein. And then, of course, later on, you want to go to high resolution. So these days, the Cryo is obviously the best microscope. It's not my favorite, but it's definitely the best. So I'm old school. backwards.
All right. I remember that CM10, and it was a wonderful instrument. Is it still in use right now? Is Zhongli still using it?
Harvard Medical School still uses my old CM10, and when I came to Rockefeller, I bought a new secondhand CM10.
Mm-hmm.
So there's another CM10 here now.
Okay. Wonderful. How has the field changed over the years for you?
It's mind-blowing to me. When I started as a student to do EM, I remember that everybody in the world who was practicing EM, who was doing EM, could still go and participate in the Gordon conference on 3D Cryo EM. Everybody was there, everybody met everybody. It was a really, really small field. And I do remember that at the time I really enjoyed doing it, but I really didn't think there would be a job for me because nobody was doing it. There were very few places that did Cryo EM, or EM in general. And so I was a little bit nervous. And then the first major breakthrough came when the field emission gun was introduced, and Bettina Böttcher and Tony Crowther at the MRC in Cambridge got the first sub-nanometer structure of a virus where you can actually see alpha helices, which was unseen before. And so that was really when a lot of places in the world started to realize that EM might actually be useful. And so a lot of places recruited people. And that was when I was recruited to my first job. And that was the first big expansion. And then the second big expansion happened more lately when these direct detectors came and basically Cryo EM took over structural biology. And that's kind of a comparable expansion that now everybody wants to Cryo EM. So from a time when I pretty much knew everybody who was doing Cryo EM to now where I really have no idea anymore who is doing Cryo EM, it's just impossible to keep up with the field. So that is certainly something that has totally changed. The other thing that has clearly changed is the resolution. That when I started, Singapore EM was really blobology. Tomography was laughed upon because nobody thought you would ever get anything out of it. And the only thing that was kind of somewhat accepted by other structural biology disciplines like X-ray and NMR was electron crystallography, because those structures you could really push to high resolution and atomic resolution. But Singapore was really just blobology. You got these blobs and people tried to figure out what they mean. And then this has changed when the fax came along, then came the CCD cameras, and more recently the direct detectors that have really changed everything. And to me, starting from a time when we looked at blobs to what you can actually now accomplish in what time is really mind-blowing to me. It's really crazy in terms of resolution, but also in terms of size. At the beginning, you could only look at big molecules like the ribosome, viruses, and when you were lucky to have a really, really big membrane protein like an ATPase or the CRC, the calcium release channel, you might be able to look at the membrane protein. And these days, you can actually look at 50 kilodalton proteins embedded in a nanodisk and get a structure. And so to me, that is just mind-blowing. It's just, I could never have predicted that this is actually happening. And what came with this is that while EM used to be a more friendly area where people were talking to each other, it has now become incredibly competitive. And I compare it to what X-ray crystallography was when I was younger. But in X-ray crystallography, you had to express and purify the protein, and then it took you a while to actually grow good crystals. And then eventually you had to phase them. And so a project took a long time, typically years, and people were competing over years. And EM, it has now happened several times to me that I became interested into a project. I started to express and purify the protein, and the structure was then published in Nature by somebody else. It's just become incredibly competitive because once you have a good protein, the time from protein to structure has become so short that it's really become challenging. And I think it's difficult for new group leaders, and much more difficult than it used to be for me. not to be eaten up by the field. And so my solution to that is to do things that are stupidly difficult or just totally different. And that leads to a lot of frustration with the people in my group. At least we don't usually get scooped anymore at this point. So the field has changed, definitely changed.
So you said that so many more people have now entered the cryo EM space and is making use of this technology. Are there any fields that are making use of cryo EM that surprised you?
So say this again, what?
Are there any fields that are now making use of cryo EM that surprised you?
Yes, there's particularly one field that I got very surprised by, and that's micro-ED, and that the chemists have started to do micro-ED. So micro-ED was supposed to start to use small crystals to make 3D structure by electron diffraction, which was started by one of my former postdocs because we all did electron diffraction at the time in my group. And so all of us always had the idea we should use this for small 3D crystals. Everybody who has done 2D crystals had the same idea. But it never worked because of the phasing problem and the multiple scattering that hasn't been really been worked out. So all of us tried and all of us failed. And so now this was a new idea to do this again. And it was basically applied to proteins. And it kind of works if you have phases from a different structure, so you can do molecular replacement. What I didn't realize was that you can actually really use this very well for small molecules where you can do direct phasing, where you don't have such an issue with multiple scattering and things like that. And so to my big surprise, it has become a big thing in pharmaceutical industry, who appear to hate to permanent structures by NMR. And it seems that micro-ED is a much better work, much better, much easier approach to get structures of small molecules. And so that has really surprised me, that I didn't see coming, but makes a lot of sense.
And to wrap up this intro, are there EM challenges that you see the field will be facing, or there are new frontiers that we will be opening up?
Well, the new challenges are the old challenges, the challenges that always have been there. And that's heterogeneity. If you have continuous heterogeneity, domains that move around and don't have defined positions, we still don't have a good way to deal with those. So we can classify and then try to get intermediate resolution structures. But a new technique, a new approach that would really take care of continuous heterogeneity, that would be a huge step forward, and the one major thing that I think is still missing in EM. An easy way to fix the problem is to crystallize the protein, and then you actually make it rigid in a crystal lattice. But that would be taking a step back. So I do think that continuous heterogeneity is still a big problem. And things that open up is certainly tomography, that you can actually look at molecules in the native environment. And so if you end up with subtomogram averaging, it's kind of related to single particle, and that certainly has opened up a lot of new ideas. And in my own interest in membrane proteins, I think what is really neat about single particle microscopy at this time is that we can reconstitute membrane proteins into nanodiscs, and we can custom design these nanodiscs. And so we can actually really expose these membrane proteins to different environments and see how they react to lipid environments. And I don't know any other method that would allow you to do that. So I think there's a lot of interest. You have 1,000 different lipids in the membrane, so there must be a reason for that. And with single particle EM, we might actually start to be able to figure out what these lipids are good for. So looking at proteins generally in the native environments, cellular proteins in the cell, membrane proteins in membranes, I think that's the new challenge or the new frontier where we can learn a lot.
Okay, great. So today's topic and why we've brought you down here, or virtually, is to talk about validation. And so what does validation mean to you? It's been changing because you've been through the field as resolution has changed. But today, what is validation to you?
To be honest, useless to some degree. I mean, at a time when you had blobs and you could not verify whether the blob actually really represents your protein or not, validation was key to kind of rescue the reputation of electron microscopy, because there's so many structures that were published that were just totally incorrect, that Singapore EM had a very poor reputation. There was a famous joke, there were 4 different structures of the PR3 receptor, and the idea was that at least 3 of them were wrong. And so in the end, all of them were wrong. So at that time, validation was difficult. There was no real way to validate whether a blob is correct or not. So at that time, things like the tilt pair test was really a great innovation to really try to have a measure whether a map is correct or not. But it didn't really last very long, because shortly after that, direct electron detectors came along. And at that point, typically structures go at least to sub-nanometer resolution. And once you're at sub-nanometer resolution, you will see alpha helices. And once you see alpha helices, you can be kind of reassured that your density map probably makes sense. And if you're at 8 angstrom or so, you start to see the pitch of the helices, at which time you not only have a good idea that your structure is correct, but you also have the handedness correct, or you can check for the handedness. So at this point, I think once you see handedness and pitch of helices, validation is kind of comes with the map. So, and of course, as you go to higher and higher resolution, you start to see big side chains, you start to see all kinds of other things. You can trust your map, at least the areas where you can see these features. Other areas, a different question, but their validation also doesn't help you.
We have several questions from the class. I wonder if I can cold call on Miranda if she can pose her questions. Otherwise, Tom will have to suffer through talking with me, and I owe him so many bags of cookies now. I don't know what to do. But if I don't hear it, so we'll progress on to at least the first.
I would actually love to see some of you guys.
Yes, if you can.
So the person who asked the question, could they make themselves visible for a few minutes?
So
welcome.
Even if dogs are, oh, okay, well, Miranda is texting me that if I can pose. We don't mind if the dogs are barking in the background. It'll be still okay.
It's just very weird to look at names on the background. It's just, this looks much more personal. So.
But if you do have questions, you could always raise your hand or type in the chat and we can supplement. But I'll proceed with the first one. So,
and let me say something. It's actually more fun if you just join in the discussion. I mean, I'm trying to make jokes, but it's much more fun if you just join in whenever you like.
Right.
I'm just mumbling, and so if you have questions, it's actually better.
Okay. Well, then, to get the discussion further along, how has public databases helped with Cryo EM? Do you make use of public databases?
So I have to admit, no, not at all, not one bit. Databases, I obviously make, well, sometimes if you don't want to go, if you have specific questions and the structure of the protein is already known, we download the structure resolution filter and use it as an initial model. But in general, we use the databases very rarely. So asking back to you, how would you use actual databases? I'm actually really curious.
So the query would also be for people reading papers and also people trying to come up with statistics on what is valid, what does this number mean, what biological questions can I pose. I think the query would be, it's not so easy to say, okay, here, I have 50 terabytes in Empire, just download it and you can reproduce what I do and you can come to your own conclusions. Um, I think we need to help people that are not in the cryo field make use of it. So that's where I see databases where we can.
So what are you talking about? You're talking about raw images, to provide people with raw images and let them try to see what they get out of it?
I think that'll be a little bit too hard now. I mean, that would be, if you're in the EM field, perhaps, if there's a question, perhaps, but right now, there'll have to be some level of trust that if I deposit a PDB, that it's not just some random model, that it mapped to model actually does make sense.
Well, that's different. I mean, now you talk about atomic models, and atomic models, if you submit them, they go through all kinds of validation. So that's not what I talk about. I'm talking about maps.
Maps.
But for X-ray, for atomic structures, there are all kinds of different ways that you can validate them. And so if you submit a map and the model that you built, your model will go through these validations. And you will not be able to submit a model that is bad or pretty bad. So I thought I'd talk about validation of maps, and for that, I don't really know why a database would help me. For structure is a different thing. That's just my view. So there was not just databases, there was something else that was in this question.
What is standard to upload along with the EM paper, the map, the raw data?
Yeah. So for that, I mean, I have to admit, I'm lazy. So we usually submit the final map that we publish, and then we publish or we submit the structure if it's applicable. If there was a structure that was built into the data, into the map, we obviously have to submit the structure, the coordinates. And that's not because we want to, that's just you have to. That's whenever you publish a map and a structure, you are now required to do that with good reason. I mean, that makes total sense. More recently, it is possible that you can actually upload all your raw data, all the images that you use to determine your structure. And while I haven't done this more recently, I actually realized that that's a very nice way to deal with NIH requirements. So NIH requires you to keep all your data for 5 years so that if any questions arise, you can show the data that you use to determine the structure. So with the ease today to determine a structure, you end up with terabytes and terabytes and terabytes of data. And so we now quite happily upload our data to Empire and then delete it on our server so we have space to make new structures. So that's really, to some degree, helpful to us just because the amount of data that we generate is just becoming overwhelming. And so that's one nice way to actually store your data and tell NIH, yes, we have the data, it's uploaded over there. But then the different things that you can upload, but map, final map, and coordinates. So in the things that we do, we often work with complexes where we don't get a high enough resolution that we can actually build the coordinates, that we can build the models. So we often use molecular modeling to make homology models for the subunits, and then we used different programs to assemble these homology models into our map. We did this with the BB zone, we did this more recently with the endogenous BAF complex. And those are not real structures. So those are, so you can actually not deposit them in the normal PDB as you would normally do. And so for that, we have to deposit them, or we do deposit them at the PDB development, PDB dev. And they have different criteria, and there we can actually deposit these kind of structures that would not make it through the normal PDB. And then it's up to you what you want to submit. I mean, some people actually submit the half maps, some people submit maps or deposit maps that didn't use any masks, so to see what the data is. But that's more or less to if you want to do that or not. What you have to deposit is the final map, and the model, and that's it.
So if I come across an entry from your postdoc that has all the raw micrographs, like the stacks, the particle picks, you're using it as an archive.
Yes,
Backup.
that's what we started to do. It's not that we're so community minded, but it's just we run out of storage space. So one solution to deal with that.
Well, brings up a question. So how much do you archive locally? How much storage space do you run your lab with?
Always too little, but it doesn't matter how much I buy, it will be too little within a year or so. So we are kind of lucky that we don't have to save the raw movies, which are the biggest stacks. They are huge. And so we have the luxury that our Cryo EM center actually stores those for us. And then for a while, we stored a second copy in our own servers, and we gave that up pretty quickly. it's just going to eat up your space, and if you have one copy, and so now we have one copy basically at the EM center, and the second copy that we upload to Empire, and that would be it for us. And because everything is just getting tight with space, I guess everybody has the same problem. So we started to delete all the intermediate steps, and we just keep the scripts more or less that tell us how we got to that, and then we can just reproduce everything if we need to. So we try anything we can to minimize what we have to store for 5 years, and after 5 years we delete because it's just impossible otherwise.
So we have a question about a valid.
So are the people here that actually ask some of the questions, maybe they can ask themselves, so they have something to do.
So then I'm going to really pick on Miranda because she has a lot of questions, even though she said she doesn't want to pose them. But
then you can't make her if she doesn't want to. Anybody?
I actually have to take off in a couple minutes, so that's why I wasn't going to pose them.
But you can talk at least once.
Okay, hold on. Let me remember what my questions were. Yeah. So I was watching the lecture video that you recorded. I think it was last year. And I was wondering if you could talk a little bit about why 2D classification might be unnecessary. I saw some people posting about this on Twitter, and then I heard you say it again in the video. So I was hoping you could talk a little bit more about that.
Yeah, so unnecessary is a little bit a strong word. So we used to do 2D classes because they improve your signal to noise ratio. So you can see the features much better of your particles. And you get a better idea of what are different orientations, what are different features or different conformations and so on. And this used to be fairly important to get an idea of what your data set is like in terms of heterogeneity, orientation distribution, and so on. And so before the new direct detectors and so on, this was really a crucial step. Now with direct detectors, you collect so much data, and if you do 2D classification, for example, in Relion, it typically puts 1 million particles into 50 classes or something like that. So you lose anyway all kinds of details of slightly different conformations or slightly different orientations. So they are not nearly as useful as they used to be, just because our data sets have become so big and they are so compressed in such few classes. But we still do the 2D classification mostly to get rid of all the junk. So we try to get rid of edges of the carbon film, of ice contamination. But now the 3D, once you have got rid of your junk particles, if you really just have your good particles, then 3D classification has now become much, much more powerful than it used to be. So you now do a much better job to do 3D classification of your particles than you used to do, or that you do these days with 2D classification of your particles. So we still do 2D classification, but just to get rid of the trash particles, and then we just do everything with 3D classification. That's the idea. Does this make sense?
Yeah, I think so. Thank you.
And also 2D classification is now actually very slow compared to 3D classification. It used to be an important step also then to calculate reprojections from the model to compare to the 2D averages. And all that has become unnecessary these days. So 2D class averages have just become much less meaningful than they used to be, or much less important. So what we use often 2D averages these days is just to give us an idea how good the data is. If we see, so first of all, we try to get rid of the junk particles, and the second thing is, do we see high resolution features in some areas of the 2D classes? And that gives us an idea what we can expect from the 3D classes. But we don't really use it anymore to look at different orientations and conformations. Any other questions that you have before you want to leave?
I actually have to take off now, but thank you for answering that one.
Okay, no problem.
So now I'll pose a question about cross-linking. So how effective do you find cross-linking, and how do you validate that you're not introducing artifacts?
Cross-linking is extremely useful in most cases. So graphics especially has really done a fantastic job at producing better maps. The problem is that it can introduce artifacts. And so somebody also asked whether negative stain has any meaning still in single particle EM. And so that's one way that we still use negative stain EM. We look at the sample without cross-linking negative stain and with cross-linking negative stain. And what we expect to see is at least some classes to the averages, so negative stain to the averages are still very useful. We expect to see some to the averages that look the same as after cross-linking. If the cross-linked sample looks completely different from the non-cross-linked sample, then cross-linking is probably not the way to go. But that's in very rare cases. It's typically in cases where you have a lot of, if the complex is very extended and very flexible, and if you cross-link, you just cross-link it into a ball, and then you can determine the structure of the ball, but you will never get to very high resolution, and the ball is never going to be particularly meaningful biologically for explaining what this complex does. But if you have complexes that are generally globular and they're just subunits that tend to fall off, graphics is a fantastic tool to stabilize the whole complex. So cross-linking has definitely improved the quality of the maps that are published or produced these days. It's a huge help. Just make sure that it looks not different than the uncross-linked sample. You just try to get a bigger population of good-looking particles. You don't want to get a totally different appearing particle. That would be a problem. And so one question that I thought was very good was how do you optimize a sample, and is negative stain EM really a good tool? And so obviously the most important parameter for you to get a good structure is to get a good protein. If you have a well-behaved, happy protein, it has become fairly straightforward to get a good structure out of it if you don't have a lot of flexibility and so on, a lot of heterogeneity. But getting this good biochemical sample, that's really what will determine whether you will succeed or not. And so there have been always, so X-ray crystallographers always use different techniques to see whether their sample looks good or not. Gels Size exclusion chromatography and light scattering are the typical tools that you use. And I can reassure you, it can look absolutely wonderful. In a gel, you see a single band. You then do a size exclusion column, you get a single nice sharp peak. You do light scattering, it looks perfect. And then you look at negative stain and it's just a total mess. So I have convinced countless X-ray crystallographers to use negative stain to look at their samples. It really, really helps. It has the most information you can wish for. It's very quick, it's very simple, and you have images and you immediately completely understand your sample. So I'm a huge proponent of negative stain. I've always been, I will always be. Every sample, almost every prep that we do ends up in the microscope, a negative stain microscope. And the first piece of equipment that I bought when I came to Rockefeller was the CM10. It's dirt cheap compared to Cryos, and you get unbelievable information. And so if you take anything away from this little lecture, if you have a chance, go and look at your sample in negative stain EM. It gives you so much more information than any other technique. And these days, a lot of people go and screen directly in Cryo EM. And it's certainly an option, it's certainly something you can do. But you have to invest much, much more time to get to the same conclusions that you get from negative stain EM within minutes. If you look at it, because negative stain produces preferred orientations, you can immediately see whether you have heterogeneity in your particles, whether there's homogeneity, whether there are moving parts. In Cryo EM, you have to collect a data set, you have to calculate 2D averages, you maybe even have to calculate 3D averages, 3D maps, until you get to the same information that negative stain EM gives you in 5 minutes. So you can definitely go directly to Cryo EM, but I think it's a waste of time, instrument time, your time, and it's wasteful. And it's much easier to just go and do a quick negative stain if you have access to negative stain EM. And typically, people that come out of my group are very frustrated if they don't have access to negative stain EM. And a lot of new places that start EM want to have a cryos and want to have all the fancy instruments, but they don't have screening instruments, they don't have negative stain EM instruments, they don't have even Arcticos to screen. And so I help people using cryos to look at negative stain, which is totally crazy. But it might still be worth to not waste more time than to have a quick look at negative stain. So I'm a big, big proponent of negative stain. The times that it has misled me, and it has misled me once or twice, are much, much rarer than the times where it really gave me enormous insights how to improve the sample. So I'm a believer in negative stain EM.
I think we had a hand raised. We do have a question.
Yeah, that was me. My question is kind of a follow-up of what you're saying. Like when you have a sample that it looks like beautiful in negative staining, and then when you freeze it, and then you don't see anything on the holes. So
yeah, so what you do with negative stain EM is just look at the quality of your protein. For that, it's fantastic. But then vitrification is a whole other beast. I mean, you have to try to get your particles into your holes. You have to try that you have different orientations. You don't want it to aggregate, which it sometimes does. So that's a different question that is equally important, but you will go through the same steps with that protein. And so then you take a lot of time to optimize your specimen preparation just to realize that your protein was total crap. So the way that my lab works is that you try to optimize every step before you go to the next step. You first optimize your biochemistry, and believe me, you can save so much time by doing good biochemistry than when you have bad protein that you try to get a structure. You will never get a good structure from a bad protein. And then the next step is try to get a good grid. And that can be very frustrating, but things have improved. there are many more tricks that you can now use to try to get a good sample than we used to have at the beginning. And so graphene oxide support is fantastic. Adding in some detergent can help. So that is not as difficult as it used to be. So there are ways that you can force your protein into the holes. There are ways that you can try to get different orientations. And if you never succeed, you can even now tilt the grid and get tilted views of your protein by just tilting. And so once you have your good specimen, then you really have to get good images. And only if you have a good specimen, prepared it well, got good images, then your image processing will give you a good structure. If any step before that is bad, you will struggle with the last step. And so for me, purifying a protein, freezing it, and then collecting data to screen for your sample is just a huge amount of wasted time, because it's very unlikely that you will get something. But as you say, once you have good protein, it looks good in negative stain. It can look pretty bad in cryo, because the air-water interface can rip it apart. That can happen. You can have it aggregate at the air-water interface. You can have all kinds of issues, but these you can now address these days by support film and all these kind of different tricks, different grids. And somebody asked me, one of the questions was, are you surprised by some way that you prepare grids that you are really surprised by? And so I have to say, long ago, when I started to do cryo, we had a test specimen. It was transferrin receptor transferrin complex. And we put it into ice, and it was just a very nice sample. And it has all these preferred orientations. And the way we sorted this one out, and again, this is now 10 years ago, so these days it's not so strange anymore. But at that time, we put a carbon film, and then the carbon film helped us not to get preferred orientations. And that was unexpected for me, because from all my knowledge, if you have a carbon film, you actually induce preferred orientations. And what was different in this case is that you had a very strong orienting effect by the air-water interface. But if you first absorbed it to the carbon, which has a much less orienting effect, you could actually prevent the particle from going to the air-water interface, and therefore we had less of a preferred orientation. It was unexpected, but at least at that time, it was still, for me, explainable. Since then, there have been a lot of techniques that have been introduced that are not explainable to me. Why a gold grid makes a protein behave differently from a copper grid, I will never understand. And so at this point, nothing surprises me anymore. So you just try, and whatever works is fine. So I think that was that. Anybody else a question? Follow-up question? Or?
So yes, I would also have a question. So if you have, let's say, a very homogeneous protein, but it has a very high degree of flexibility, it would probably still not give very good data. What are some ways how you can improve or evaluate, you know, like if you have something that's very flexible, maybe has some intrinsically disordered regions, that the structure is correct? And
so if you just have flexible loops, those will just be usually averaged out typically. So that's not your problem. The problem typically is if you have big domains that don't have defined orientations, but they are just flexibly tethered and can go in all kinds of orientations. And that's a problem that hasn't really been sorted out still to this day. So the typical things we try to do if we deal with something like that is to see whether the protein has activators, ligands, substrates, anything that interacts with it, and possibly then prefers one conformation over the other. So what I found that one of the things that I start to believe in is that if you have a biological system and you have a flexible domain, very often you have one functional state where you have a defined structure. And all the other structures which don't depend on this defined structure can have all kinds of different orientations. You don't care, it just doesn't need to be that defined structure. And this defined structure can either be inactive, and so to keep this protein from being active, you have to take this domain in so that it doesn't do anything, or it can be an activation that this thing has to be released from the core complex so it becomes active. But quite often what I found is that you need one defined state for protein to be active, and the rest can look whatever it wants. This is not a general rule, but it quite often applied to what I found. And so what we typically try to do is to find anything that interacts with this complex: nucleotides, DNA, RNA, ligands, anything you can think of. Another trick is you can try pH. You can change the pH. That sometimes changes how flexible domains behave. But if nothing of this helps, then you have a real issue. And then typical, you just determine the structure of the core domain, and then you try to determine the structure of the flexible domain separately at high resolution, either by X-ray, NMR, or whatever you have. And then if you can collect enough data, you might be able to get low resolution or intermediate resolution structures of different conformational states that you can then use to dock the high resolution structures into. And that might be the much more practical approach than to try to get high resolution structures of all the different conformations, which might be impossible. There's just still, to this day, there are no good approaches to computation that try to deal with this. People have tried for a long time, but there's just, in the moment, there is nothing. And then if you're really desperate, you can try to introduce cysteines and try to make the sulfide bonds to really nail it down. But for that, you actually already have to know quite a bit about your protein. And if you're even more desperate, you can do graphics or glutaraldehyde and just glue it down. The problem is it can be glued down in all kinds of different ways, and it might not help you to get to a high resolution structure either. So this is the problem that nobody has really figured out yet.
A question I want to pose to everyone here, and you can type your answer into chat. If you had to solve a reconstruction, what would your resolution range goal be? Because I remember talking to Tom one time, and he had wonderful data in stain, but the reviewer said, well, it's not in cryo, so I don't believe it. And then now I think the question becomes, at what resolution? And we might have to keep on pushing. So if you can type in chat, if I gave you a cryo EM paper, what resolution range or max would you feel comfortable in believing any claim, or do you have any preconceived notion? I don't see any answers. Should I start up? Oh, there we go. Now we got it. 10. Wow. Okay. Here's 2 to 4. People are generous. So then, Tom, have you encountered resistance, or do you have opinions on how the field has shifted to requiring this strange number called resolution before certain data is believed?
Stupid reviews have existed for a long time, and it hasn't improved. I hate numbers. I think they're the most used, not the most useless, but it doesn't matter what your resolution is. It matters what you can see in your structure. And if you have X-ray structures of all the different subunits of a complex, and you can get the complex even just at 20 angstrom, and you can unambiguously dock your proteins into your density map, 20 angstrom is fine. It's all you need to know. You just need to know the envelope, and you can answer a biological question. As you go higher and higher in resolution, you can start to address more and more questions, and you can be more and more certain of what your structure is correct. However, this focus on you have to have a certain resolution to be publishable irks me to this day. If you have, what very often happens, you have, let's say you have a data set of 1 million particles and you publish a structure at 3 angstrom. And then you go into materials and methods and you realize that this structure was actually done from 50,000 particles. So now you have the 50,000 particles. How representative is this of a particle population of 1 million particles? And I have many more issues when people give me something like this. I always ask them, what is the rest, the 950,000 other particles, what do they represent? And to me as a biologist, that's where really a lot of the interesting information comes from. How does the molecule move? What is, and I don't really care all that much if I have one structure that gives me what the atomic model is. I start to find it much more interesting how do the different domains move with respect to each other and actually really provide biological insights and mechanism and things like that. But this is, anybody who has submitted paper knows that it's sometimes difficult to deal with preconceptions of certain reviewers. And I think one problem of the field is that now there are many people who have only gotten high resolution structures and become independent group leaders, and then have as a preconceived notion, a structure has to be 3 angstrom, otherwise it's not a structure. There are lots of people who were trained by ribosomes, and ribosomes always go to 3 angstrom, but it's kind of trivial. While you can learn much more from a smaller protein, not maybe just at 4 angstrom, but giving it a new fold or whatever. So since I'm in the field, and for a while I had the structure with the highest resolution, I had 1.9 angstrom resolution a long time ago. And it never mattered to me. It's not important. It's important what you see. In that structure, you could see interactions with lipids, which we couldn't see otherwise. But that's what matters. And so some of my nicest results to this day are negative stain because it revealed activation mechanism of an integrin at those this stuff, that you can actually see that an integrin gets activated by extending the molecule. And that you couldn't even see in the X-ray structure of the compact molecule. And that was really a deep insight into the mechanism of this molecule. So personally, I don't care about what resolution number is attached to a structure. I always only care what can I see, and what I see, does it address the biological question I'm trying to answer? So one of the questions was, in the future, will we always need 1 angstrom? I think at that point, I would just give up. It's just pointless. That makes no sense anymore. So numbers are useless. What you can see and what you can answer, that is what matters to me. So all these questions to me are correct. At less than 7 angstrom, you can see helices. You can see how the transmembrane alpha helices are arranged. At 10 angstrom, you can see in a complex how the subunits are arranged with each other. At 3 angstrom, you can see side chain. At 2 angstrom, you can see water molecules. If you're interested in a mechanism how proteolysis works, where you need the water molecules. So all these answers are correct for different types of questions you might want to address. And then you just have to fight with the reviewers. But you always have to do that. So it's just nothing new.
Well, I guess circling back to reviewers, I wonder how many reviewers would want to see raw data. Sometimes reviewers want to see maps, but would that even be a reasonable request? Because it's not like a reviewer would be able to reprocess. Well, they might be able to.
No, and you can, all this comes from olden days when there was a lot of issues with what you would be able to interpret. So the one example that I presented in this is this 2 angstrom structure of GP140, of GP160, which was a total ridiculous map where you could actually see glycosylation come from the transmembrane domain. So if you have a map, you immediately see that this cannot be, and you can look at the map and you see that the map is totally meaningless and it's all just noise. So at olden times, it would make sense to look at the map, and you will also see in the map whether you have strong preferred orientation, you have very anisotropic resolution in the map, and so you have to be careful what you interpret or not. So if you're very insecure about something that authors interpret and you just want to know whether this is really in the map or this is just fantasy, I think it's okay to ask for the map. I rarely do it, except I have real doubts that. But these days, with relying on CryoSpark and all these programs, you usually get decent maps. And one would hope that scientists are honest enough that when they have an issue with the map, that you have preferred orientation, you have anisotropy, you can still use the map, but you have to be more careful how you interpret them. And so we write this, we have preferred orientation, we have anisotropy, so we didn't build side chains, we just have to fold or something like that. So that's the responsibility of the scientists to really interpret maps to the degree what they're allowed to interpret and not further than that. And if you as a reviewer have doubts that you think they went overboard, then looking at the map can help. Raw data I don't think will help, because raw data, you're not going to go to look through 100 different images and see what they look like and calculate CTFs and things like that. But map, I think, is reasonable if you're in doubt. Obviously, it irks me when I'm asked to send maps. It's okay.
This is a kind of general question, but whenever you're asked to review any structural work, do you have any specific things that you always ask for, or is it on a case-by-case basis where you read the paper and you think, okay, this is something that clearly maybe they should consider regarding validation?
I think these days it's almost a given what you present if you show. So you show a raw image, which is basically meaningless because you always show one of the best. So, but you look at the 2D averages, you do look at the workflow. Does it make sense what they did? Did they introduce bias by using whatever it is? And then you look at the Fourier transform, the Fourier calculation. You do look at the fit of the model to the map. So it's almost a given. But what I realize is that these days, more and more, I get papers from people who have just started to do EM that show nothing. That just show a raw image and then they show you the structure, not even a map. And that is just because it had become so easy. You can basically take your protein, send it somewhere. Somebody does the structure for you. You get the structure. You don't understand what you're supposed to do, what problems are. And I typically always write in my papers how many particles we use to get the structure to say whether it's representative or whether we have just a substructure. And extreme examples of that are actually these amyloid fibrils, where often you use 0.001% of the segments that you picked to get a structure at high resolution. So that's one structure, but I bet my life that there are 100 other structures in this data set that you will never see. And so that's actually a reason why I stopped working on this stuff, because I just don't believe anymore what I get. It's one thing, but who cares? That's the most defined structure, but it's probably the biologically least interesting structure. So, but it's complicated. But if you look at papers that come out of EM labs, you can see what people show, and that's generally what you're supposed to show. But it's become difficult to judge papers, I have to say. Yeah, but it's probably easier than it used to be. So there's overall, once you have decent resolution, it's much easier to trust the paper than in former times when you had the map at 20 angstrom and it could be anything. And also validation methods that have now been used that you actually cross-correlate or do correlation between the map and your model. All these things have become very, very helpful to actually really assess the quality of your model. All this didn't exist in prior times.
Thank you.
Any other questions that I didn't address?
Well, if I can ask a question, I'd actually be curious how you think in thinking in the future. How far do you think can you automate, or can you imagine that this can be automated, the process? Because right now you have decision points that are need human experience, but if you compare it with crystallography is pretty much automated if you want to do it brute force, and that's just further ahead. And I wonder if you think it can be automated, and that would enable like the substructures, as you call them. You could determine more structures in parallel potentially if you can automate the process.
So I believe it will be automated sooner or later. And that's what Ed and his colleagues are working on. With artificial intelligence, deep learning, deep algorithms, I think it will be possible. It will go into this direction. But it will always need a human to check at least what happened in the algorithms. So I do think so these guys at NYSBC, they actually put in the grid into the microscope and end up with a structure in the end, right? Pretty much without intervention at this point.
There's some intervention. It's not necessarily the right structure. It's whatever you put in, you get something out.
But let's say the proteasome you get without any intervention, right?
Very little.
So basic structures are pretty much already totally automated. The more complicated probably will take longer to fully automate, but might also get there. And then I have to find something else again. it's almost getting boring. If you have a simple protein, the challenge is almost gone. So it's much more interesting to work on a complicated problem, because then at least it's not just cranking. It's really trying to do something special. So I always laughed at X-ray crystallography because it was always push button. And EM was still twiddling around. It took us 10 years to get a structure, but it was fun to do it. And now we can get a structure in a day, and it's no fun to do it anymore, at least for me. It kind of took the fun out of me. And the response is now you can do more biology. Yes, it's true, but it used to be fun and challenging to get an EM structure. And it's not as much fun anymore, at least to me. So I do believe eventually everything will be automated, and then we can all look for a different job. It's kind of two-sided to me, two-faced edge. I really used to enjoy doing things by hand, and a lot of this has gone away.
Well, my impression would be if you just move it up by one dimension and start working on whole cells, like you're back into like, at least that's my take.
That is correct. Tomography is the new Wild West, but it takes me always a very long time to move from one field to the next. I've done 2D crystals long after most people did single particle, so I'm always behind the curve. And I decided not to go into tomography. I'm more interested in looking at mobility, flexibility, different conformations. That's the challenge that I'm interested in. And my personal challenge is really lipid protein interactions. Membrane proteins and nanodiscs is what I'm interested in doing.
Well, that brings up actually one more question now, because I think about mobility and flexibility a lot. I mean, do you see a way where we can get this to a place where we can end up with a movie of the molecular dynamics of your complex? I mean, from the data, I mean, it moves in this direction in, I think, good cases, but is this something that may be happening for anything that's flexible?
I think it might happen. But it will still be a long way. There are new combinations between molecular dynamics and EM data that actually might eventually really lead to a real molecular movie of what happens, not just a guess. But for that, you will have to be able to really bring out very minor populations in your poultry population. And that is a challenge to find small populations in large data sets. But there are now more and more programs that are trying to do that, and they combine this with artificial intelligence, deep learning, molecular dynamics. And I think it will go there. And that I think will be very exciting. That will really start to address a lot of really mechanistic questions. But there will be many steps in between to get there. Any more questions? Nothing has to do with validation, as you might have noticed.
Or we can flip it. Tom, do you have any questions for everyone else?
Me?
Right. If we're constantly asking you questions, isn't it fair that you ask questions?
How many of you have actually done negative staining? Just out of curiosity. Oh, still quite a few. Okay, excellent.
Actually published a paper with negative stain data, which I feel like is rare nowadays.
Who said that?
Oh,
excellent. Very good. What did you do?
we were working on, actually with one of your colleagues at Rockefeller, Jim Hudspeth, on mechanotransduction in the inner ear. There was this very long sort of filamentous-like protein called protocadherin 15.
And you know that he was for the longest time not able to image it.
Yeah.
We then showed him how to do negative stain, and then it worked. You can ask him.
Yeah.
So I know the protein.
Yeah. So that's we did. Obviously, this was with single particle stuff. There's another grad student doing crystallography and then some things like analytical ultracentrifugation to basically try to identify the dimer interface, because there are these repeat domains in the protein, and we didn't know which one was holding the dimer together.
Very nice.
But the cryo didn't go to high resolution, but the negative stain gave us a really nice, at least like domain level resolution map, which actually gave us the sort of pitch of this helix, which gave us lots of information about how long it might be if you were to unwind it, which correlated really well with the biophysical measurements Jim Hudspeth's lab was getting.
Yeah, Jim only asked us how we do it, and then we showed him, and then we never saw him again. But I'm glad it worked out so well.
Yeah, it did.
All right. No, I don't have questions. Who struggles with unusual problems in Cryo EM? For who is the process smooth and easy, and who ran into any kind of problems? Okay, who had problems? Not that many. So field has come a long way from when I started there. We all had problems. So yeah, that's a good sign.
I still work with lots of very long, flexible proteins that are usually pretty tough to get high resolution structures of. So
whether we?
I do.
You do?
So,
okay.
Yeah, I always struggle.
Yeah, so yeah, good luck with those. Yeah, those are not easy. So it depends what you work on, but we had we worked on tethering complexes, and eventually we gave up because there's just too many flexible parts. And for a while I was worried because Shaw said he was going to use this to work on flexible complexes. And so that was another reason for me to give up, because if Shaw's can do it, but he never managed to. Shaw's also gave up. So flexible proteins are still a big problem. It's not easy. All right.
Okay, well then we can wrap up. Any words of encouragement rather than what is fun? If you say that Cryo is not so fun, if it's not fun, shall we open up a dessert shop? I can make cookies for you. You can make some chocolates.
No, find what you're interested in that is not cookie cutter. I mean, literally, I could not in my life determine the structure of a ribosome. I would rather kill myself. I just could not do it. But things that what I think EM is great at is looking at different sort of conformations of a population. I think no other, well, NMR can for small proteins, but EM is really unique in that you can look at these different conformations. So that is for me interesting, and the interplay between membrane and membrane protein. That's for me, that keeps me. But the actual structure determination is not that much fun anymore. It's much more fun to think about ways how to tackle these problems. But I think what is different these days is I used to be an electron microscopist. And that breed is going to die out. You're not going to be an electron microscopist anymore. You're going to be a biologist who is interested in a certain biological problem, and then you use electron microscopy as one of your approaches to address your question. But what I used to be, that you just are an expert in Cryo EM, that's going to totally go away. So I'm a dinosaur in this kind of way. And I now more and more go into biology and use Cryo EM as one of the tools that I use. So my advice for everybody is, don't become a technologist in terms of EM. Those times are over. The problems are more or less solved. Just focus on the biology and use what EM can provide you as a great tool to answer your questions. So for me, it's frustrating because I used to be an EM specialist and I'm no longer needed, basically. So now I have to reinvent myself. But I can still use EM to look at things that other people wouldn't look at. So that makes me still. But you remember 10 years ago what it was like to do a Cryo EM structure. It was a different beast than it is now. But we, as I said, it took us 10 years and we got to 7 angstrom and we were very excited at that point. So what you get these days is so much better than what I used to get. And really, every day, somebody else determines another structure that just cannot believe that it was possible. I mean, things get smaller and smaller and more complex. I mean, it's mind-boggling for people like me who started out 30 years ago. So I might just not have the imagination what to do, but there's so much you can do these days with Cryo EM. It's really remarkable. And then there's always tomography. I just haven't gotten there yet.
Okay, wonderful. We can stop recording now.